Welcome everyone for today's seminar. So my name is Kiko. As Christina said, I work at the Laboratoire d'Oceanographie de Villefranche-sur-Mer, and I work on the Make Our Planet Great Again project, Tropical Atlantic Deoxygenation, Gateway Dynamics, Feedback Mechanisms, and Ecosystem Impacts. And today I want to talk about Oceanic Oxygen Minimum Zones, Death Zones, and Refuge. And this work is also co-funded by the EU project TriAtlas, South and Tropical Atlantic Climate-Based Marine Ecosystem Prediction for Sustainable Management. And much of what I'm showing, as Christina said, is also related to my previous work at GeoMar in Germany and in the Collaborative Research Center 754, Climate Biogeochemistry Interactions in the Tropical Ocean. Okay, so what I want to do today is to some extent give you the background on oceanic oxygen minimum zones. I want to explain what these are, why they are where they are. I also want to relate a little bit or explain a little bit what consequences oxygen minimum zones have, and then I also want to show you more of the work we are doing in the MOPCA project that is ongoing. So what you can see here is a map of the oxygen concentration at 300 to 500 meter depths in the global ocean. And you can see, so the color indicates the amount of oxygen. Warm colors mean, or yellow and green colors mean there's a lot of oxygen, and this blue and violet colors mean there's very little oxygen or even no oxygen. And what you can see is that there is a specific distribution here. We especially have little oxygen here at the eastern boundaries of the continents, but also in the Arabian Sea and the Bay of Bengal. And yeah, I want to explain, first of all, how this comes about, why they are where they are. If we talk about this, we have to talk about oceanic oxygen dynamics, and we have to consider the different processes that can change oxygen in the ocean. These are oxygen uptake, oxygen transport, and oxygen consumption. And these happen in different places in the ocean, and that's why actually, if we understand these processes, then we also to some extent can understand the distribution of oxygen in the ocean. So oxygen uptake takes place mainly at or exclusively at the surface of the ocean. We have, first of all, the mixing, or the atmosphere contains a lot of oxygen in comparison to the ocean, and so at the ocean atmosphere interface we have interaction there, and we have oxygen just dissolving in seawater. And also when there is a lot of wind, for example, we have the water mixing, and that's how oxygen is brought into the ocean. Then we also have photosynthesis happening at the surface. That's the algae, for example, that use light and CO2 to produce biomass, and as a byproduct they also produce oxygen. And also this process only happens at the sea surface, where the light penetrates the ocean. So oxygen uptake takes place at the surface. Then you can have waters being submerged in the ocean, and they can be transported along within the ocean, and that then, so to say, is a distribution pathway of the ocean, of oxygen. And while it is transported, you can also have consumption. And the consumption is due to, for example, dead biomass being respired by bacteria. Of course, you can also have fish that consume oxygen, or zooplankton, which is what I'm dealing with. They all consume oxygen. And that happens, of course, throughout the transport of the water mass. And that means the further away you are from the source where oxygen is mixed into the water, the lower normally the oxygen levels are. And so what I'm showing here is now a scheme of the global circulation that we find in the ocean. This is called the thermohydroline circulation. And what you can see is that to some extent this thermohydroline circulation actually starts here in the North Atlantic, where we have where it's in winter it's cold, and you have sea ice forming, and that means that actually water becomes more dense and sinks and penetrates to the deeper ocean. And then that water can be distributed throughout the oceans. And so what we can see from this is that, first of all, we should have high oxygen levels here at in northern regions, and to some extent we have lower oxygen levels further away from this source region. Yeah. And yeah, that's one reason why we have one mechanism to explain the oxygen concentrations in the ocean. And you can see here, really, as I said, we have high levels here, oxygen concentration, and the further we go away, so to say, from this region, we also find lower oxygen levels. What we also have is another important mechanism that creates these oxygen minimum zones, especially at the eastern boundaries. This is a process called upwelling. We have in these regions, predominantly here in the northern hemisphere, northeasterly trade winds, and in the southern hemisphere, southeasterly trade winds. And these, so to say, go along the coast here, for example, of Africa. And this actually creates something we call an Ekman transport. That means that water, there's wind-driven upwelling. So water is coming up here at the eastern boundaries from depths. And this upwelling actually then also generates a high productivity in this region. So the water that comes up from depths actually contains a lot of nutrients, and these nutrients are used by phytoplankton, by algae in the water, to actually conduct photosynthesis. You have a lot of organisms in the water here, and you also have something that we call marine snow and also fecal pellets that organisms create. They sink down in the water column, and this then actually generates also high oxygen demand at mid-water levels. And so overall, we can say that these oxygen minimum zones we find here at the eastern boundaries, that they are formed due to this high oxygen consumption that happens here in these regions, but also because they are far away spatiotemporally from the deep water formation areas. So water here might be something like 10, 20, 2 years old, whereas water here at 300 to 500 meter depths in the oxygen minimum zone of the Atlantic might be on average 80 years old. And here water in the Pacific is even older. And that's one reason why we actually have very intense oxygen minimum zone with very low oxygen levels here in the Pacific. Okay, so that's the explanation for the spatial-temporal distribution. What we are, of course, also interested in is what's the temporal change here. So does this change over time? Are these oxygen minimum zones changing? And what we know from observations that somehow are reliable since about 1960 is that actually, yes, for example, in the tropical Atlantic, we are losing oxygen at different depth levels. So this is from work by Sunke-Schmidtko et al. From 2017, which shows that, for example, at 3,000 meter depths, we have a decline of oxygen of about 5 micromole per kilogram. So that doesn't sound much, but we'll get back to that. And what you can see also is that actually in the shallower depths, for example, at 300 meter here indicated by green, we have a decline from something like 5, of nearly 10 micromole per kilogram since 1960. Yeah, so actually the tropical Atlantic as a whole has lost about 2.2% oxygen in the last 50 years, but that is integrated over the entire water mass, and so down to 5,000 meter depths. And yeah, for example, at 300 meter depths, you see that the trend is actually stronger than the trend at 3,000 meter depth. So this oxygen loss that we find in the ocean has not yet reached the deep sea, and that's why overall we don't see such dramatic loss. But globally, we also know that this is 2% oxygen that the total global ocean has lost since 1960. And yeah, if we look into the future, then we expect this to actually get worse, that the ocean probably will lose further oxygen. So what you see here are predictions from different CMIP6 models. So that's biogeochemical models of the oceans, which first of all try to model or predict, generate the past, and you can see that they do quite a good job here. So that's the past records we have, and then they actually try to predict the future. And what you see here is, on the one hand, the sea surface temperature plotted here on the y-axis versus the year, and you can see that depending on different scenarios, the ocean will warm to certain degrees. And because I've explained at the beginning that oxygen actually is mixed into the ocean, and because seawater, warm seawater can hold less oxygen than cold seawater, we also have a pretty direct effect of this sea surface temperature increase on the predicted oxygen. So the oxygen levels in the ocean will also decline further if the ocean warms, which we expect due to global warming and to the increase in CO2 in the atmosphere. Yeah, so overall, we expect oxygen to decline in the ocean. Exact predictions are difficult, and locally, we expect locally different responses, for example, due to wind and temperature forcing. So when, for example, global warming leads to an increase in wind forcing, then this should also lead to, at a certain location, then this should also lead to an increase in oxygen supply at that location. But overall, we expect it to decline. And we know that about 50% of this ocean deoxygenation can be, that we have observed in the past, can be explained by global warming. The other 50% are likely due to biology, and that's actually what I try to understand. Okay, so does this matter? And to talk about this, I want to start with a picture that I have taken myself and take you up on a mountain. This is actually at about 5,200 meter height in Peru. This is a picture I took myself on the so-called Rainbow Mountain. And yeah, that was actually quite interesting hike I've done. That started about 4,300 meters, and it was about 20 kilometers to get to this location, to this Rainbow Mountain. Yeah, when you start this hike, you think, okay, everything is nice. You're well adapted to this, but then at some point, maybe some headaches start to get to you, and you don't feel so well. And that is due to the fact that at these heights, oxygen levels are also already quite low. And if you're not well adapted to it, then you might suffer. And that is what I actually did. And at some point I thought, okay, I don't really want to go on. I want to turn around. But luckily, the locals know about people like me, that some people have troubles with the height and with reduced oxygen. And so I could hire a guide to actually bring me up there. And so I had to enjoy the horse ride up there. But then at the end, actually, it's the horses cannot go up the last few meters, about 200 meters, and then I had to walk the rest. And at this height, there's really very little oxygen, and so I had to stop every 2 meters and regain my breath. So for me, this was really an interesting experience to really see, okay, this is how low oxygen level impacts myself, and how oxygen how important oxygen is. I mean, we all know that, but we rarely have the opportunity to experience it. So for me, it was more or less, the sign was very interesting. So that's my personal story about oxygen tolerance. So I have a little bit lower oxygen tolerance, it seems. But this is, of course, also interesting in the ocean in general. And what I want to show you here is the some results from colleagues from Kiel who have investigated how decline in oxygen levels might impact billfish. So fish like here, this big swordfish, or also big tunas, which need a lot of oxygen to perform. And what you can see here on the left side is the distribution of oxygen in the water mass and the dive track, or the dive track of a swordfish. So this is a yellow line here, which was recorded with a tracker that was attached to the swordfish. And what you can see is that the swordfish actually did evade these red areas, or did not penetrate into the red areas. And then when the oxygen levels got higher in the seawater, then they actually went down and also did hunt in deeper water layers. And so what the colleagues investigated is they checked how actually the level, this 3.5 milliliter per liter threshold level, how that changed over time in the tropical Atlantic. And what they found is this, that actually mainly here the regions where oxygen is below this threshold has showed. So this mostly you see here on this map that most of the map is red. That means that each year the level at which you find 3.5 milliliter per liter oxygen actually has come up. And this means that the habitat that these swordfish, but also other billfish, as said, like tuna, that they can use has declined, because they can just not use these layers. And so we see that the habitat declined by 15% between 1960 and 2010. Okay, so I'm actually not working on billfish. I'm working on other topics, and I want to motivate that a little bit. So as I've explained in the last few minutes, oxygen levels globally decline, and they also decline in the tropical Atlantic. I've talked about the billfish, tuna, swordfish that actually are impacted by that. But you can imagine that these actually feed on smaller organisms, like smaller fish and squid and some crabs or krill also. Some small crustaceans, and these again feed on even smaller crustaceans and other small organisms that we find in the ocean. And finally, all this is based on phytoplankton, as I've said, that for example fixes sunlight. So this is the trophic pyramid we find in the ocean. What you can see from it is that, for example, the abundance of fish that we have in the ocean also depends, of course, on the one hand on the productivity of phytoplankton, but also on the amount of zooplankton that we have. So zooplankton feeds on phytoplankton, nekton feeds on zooplankton. So this trophic chain is, of course, important to also explain, for example, the distribution of fish in the ocean. And so what I'm interested in mainly is how zooplankton is impacted by declining midwater oxygen levels. And that's especially interesting for the upwelling regions, where we also have a lot of fishery going on. And these fish, as I said, they depend on zooplankton availability and also on phytoplankton availability. So overall, we actually have to understand better how the changes in oxygen might impact the entire ecosystem. And my focus then is on zooplankton. Another reason to work on zooplankton and why that is impacted by declining midwater oxygen levels is the fact that zooplankton is not only important for this horizontal, so to say, transfer of carbon from phytoplankton towards fish. It's also very important for the vertical transfer of carbon towards the deep sea. So zooplankton, for example, as said, they feed on phytoplankton and microzooplankton, and they create fecal pellets which sink. And these fecal pellets then take carbon to depths, and this is an important aspect of the global climate regulation. And they also migrate to depths, and also these migrations, because they respire, for example, at depths, they also bring down carbon to depths. And the oceanic carbon flux that is only due to this pathway here, to the organisms migrating to depths on a daily basis, that is about 30% of the global carbon export in the ocean. So there are two very important functions that zooplankton has. They are the food, so to say, for higher predators, higher trophic levels fish, but they also are important in the carbon export to depths. And that's the topics that I'm dealing with. I've talked about zooplankton, but I want to also explain a bit more what this is. So the term plankton was actually defined by Victor Hansen, and he actually was also the first to build, to start quantitative work on plankton distribution in the ocean. And he defined plankton as organisms that cannot evade a slowly towed net. And zooplankton, the organisms that I'm working with, which you can see some of them here, these are the heterotrophic parts of the plankton community. So those that actually feed on other organisms, like zooplankton feeds on phytoplankton. And what you can see here are a few examples of zooplankton. You have some cnidarians, some medusae. You have here some pteropod, and you see also a few crustaceans, like here is a small crab, but this is also a copepod, one of the most abundant groups we have in the ocean, with which we work a lot. And so that's some examples of zooplankton organisms. You can watch or take a look here at Christian Zadeh's website, which has some very beautiful images of zooplankton and other oceanic organisms, if you who are interested. So what are we doing to actually investigate zooplankton? We are still using plankton nets. That's still very important, but we actually have nets now which can sample different depth layers. So that's why this now looks a little bit more complicated. So with this net, for example, we can sample 5 net layers, depth layers. And we also use no longer, we don't count all the organisms that are on these in these nets. We actually do scan the content that we find in our nets and take digital images of these. And we also use a so-called underwater vision profiler, which was developed here at the Laboratoire d'Oceanographie de Villefranche by Marc Picheral and colleagues, which is actually a camera system which we can deploy on so-called CTD. This is a system that we send down to 6,000 meter depths if the seafloor is so deep, with which we, for example, measure temperature and salinity, but also can retrieve water samples from depth. So that's a standard oceanographic tool that we use, and we can then put the underwater vision profiler onto this oceanographic tool and also deploy it with it. And while this instrument goes down, it takes also images of the water. So what you have is that actually here in this metal steel cylinder, you have a camera that looks down onto this plane here. And this plane, these 2 tubes here, they are LED lights which shine towards the middle and create an illuminated volume. And this volume is image with a rate of 20 Hertz. So that means 20 images per second. And so while the CTD goes down with something like 1 meter per second, we take 20 images of the water in this volume here. And so the water is, of course, exchanged while the CTD goes down. And so we get a profile of all the organisms and particles we have within this volume. Okay, so this is how some of the images look like. The camera is black and white or a grayscale camera. The light we use is actually red, and that's why we don't get any color images. But you can see that we get beautiful images, and a lot of detail here. So this is a foraminifer, this is a copepod, this is actually a polychaete, and this is, for example, medusa. And we get them in situ, and this is a marine snow particle. And we get these in situ, and we can therefore actually relate them to environmental conditions, for example. Okay, and yeah, as I said, we can deploy that on a standard tool, this CTD, which we can deploy on cruisers with research vessels. And you can see here that the data distribution we have now. So we have many, many profiles of this underwater vision profiler, which we can analyze. We also now use, thanks to the MOPCA project, we also use another imaging system, which is the Underwater Vision Profiler 6. This is much smaller, and we can, for example, deploy it on moorings. So we have deployed it for nearly 2 years. Now at a location here, 23 West in the Atlantic, but also at another location. And what we also do now is that we also deploy it on so-called BGC Argo floats. That's autonomous tools we can throw in the ocean, and they measure a long time. They go up and down in the ocean, and thereby also create these profiles that we are interested in. And they can transmit the data that we are interested in via satellite. And so this is a track of one of these floats that we have deployed. Each dot here now represents one profile that we have generated with this robot, so to say. What you can also see is that there are some gaps in this nice profile or in this nice track, and we really had some problems with this float, and therefore we have some gaps in the data distribution, and we actually decided to ask the French vessel Thalassa to pick this float up. And so last week on Wednesday, I had some very interesting time to watch online whether Thalassa can actually make it to pick up this float. The problem was a little that there was only a very small time window where actually Thalassa could reach this float before sundown. And so for me, that was a little bit like a Pac-Man reminder here, seeing how Thalassa actually got closer to the float. And then finally, the float signal disappeared, and I got some good news from the vessel that they actually picked up this float, and that the camera and everything was in good shape. So we are very happy that we got this float, and we'll be looking at the data in the near future. Okay, that's maybe a little bit excursion to the normal life of a biological oceanographer, bringing our tools and trying to get them back as well. I now want to just quickly explain what we see with this data, and why I refer to the oxygen minimum zone as a refuge and a death zone. So what we, for example, can do with this data now is we can pair it with the environmental data we obtain also with the CTD. And I want to take you first to the oxygen minimum zone of Peru, where actually we have zero oxygen between about 100 to 350 meter depth. So that is what you can see here in blue is the oxygen in partial pressure, and red is actually the temperature. And you have here on the y-axis the depth. And you can see that there's no oxygen at this depth. And what we can do now is we can also look at the distribution of, for example, krill. That's a bit larger crustaceans, like something like your small finger sized, about that, and copepods. And what you can see is what I've plotted here is also the distribution, the relative abundance of krill at daytime and nighttime. And what you can see is that this krill here actually migrates to depths at daytime, and actually really can stay at this depth level where we have no oxygen. And it goes up to the surface at nighttime. And this is due to the fact that they try to hide from predation. So they go to the surface at nighttime to actually feed, and they go down to depths at daytime to actually hide from visual predators like bigger fish, etc., that would feed on them. Yeah, and you see that the copepods don't do the same, so they are more inclined to stay in the oxygenated layer. And if we, for example, plot that the relative abundance here versus oxygen, we see something quite astonishing. That is that although it looks like a lot of krill actually goes to the oxygenated layer, they actually really go more to slightly oxygenated layers. This is something that astonished us quite a lot. Whereas, for example, for the copepods, we see that they mostly prefer the oxygenated layers. What I want to say with that is that in this case of Peru, the krill actually uses the oxygen minimum zone as a refuge. We know that also from other work, for example, from ADCP data, which actually here symbolizes or shows nicely the diel vertical migration of these organisms. They stay at the surface at nighttime and they go down to depths at daytime. And so this is really a evasion mechanism they use. And because the krill can actually do that, they are incredibly abundant off Peru. So it's really a keystone herbivore and prey of the Humboldt current system, and a very important aspect that they can do that. So if we now take a look at the same story or the same data from the Cape Verde region, then we see that we have here the oxygen levels are not so dramatic. So that's shown here on the top left. You have here depths versus oxygen, and you can see that the oxygen levels in the Cape Verde region right now maybe reach 40 micromole per kilogram at depths here. And if we again take a look at the distribution, then we see that actually, for example, krill uses the oxygen minimum zone also to some extent at night. They also migrate up and down. And they both krill and copepods, they have this shape of oxygen preference. And so for both of them, the oxygen levels are not so problematic. So here, the copepods also do thrive in the oxygen minimum zone. What we don't know to some extent is how the future looks like, because what we can from this data only gather is how they are distributed right now. But if we expect oxygen levels to decline, then we also have to study low oxygen situations. And that is what we do in some, or have done in some experiments. So we, for example, took the copepods and krill from net catches and put them into small bottles and actually measure how the oxygen within the bottle declines over time. And from this data, we can then determine the respiration rate of the copepods and the krill, and we can also determine whether they actually, to some extent, get into stress, like I got into stress on this mountain. Yeah. So what we can do with this data is we can plot, for example, the respiration rate here on the y-axis versus the oxygen concentration in the water. And what you can see here in general is that there is a region where you don't see much changes in the respiration rate of the individual krill. And then there is something like I have depicted this here with these lines, is when actually the krill dies, or actually at least reduces the respiration rates dramatically. So what we see here in principle is that krill and copepods, this is a copepod, this is a krill, shown here, that they actually reduce their respiration at some point, what we call the peak rit, and that they actually then at some point below that even die. Yeah, and so this is, so to say, the oxygen tolerance threshold here of these two species determined. And for example, if we then compare it, what we find for the species to the oxygen levels in the water column, then we see that the oxygen levels in the water column are maybe something like 5 kilopascal here at 300-400 meter depths. But the krill, for example, its oxygen threshold is 2.4 kilopascal. So it actually can still has still some breathing room, so to say. So they can still use these depth levels to migrate to and to thrive there. Okay, so this is now from a location I have called ZOMZ, which we have investigated intensively in the past. And yeah, I want to quickly compare data that we have obtained in this region to other regions in the Cape Verde area, where oxygen levels are a little bit higher. And what we've done, as I told you, we use this HydroBIOS multinet, which can sample several nets to actually determine several depths, to actually determine the biomass of organisms in these different depths. We then scan the net content, as I've already explained, and we get the images you can see here on the right. And then if we actually segment it and get the size, we can get to biomass. And if we also classify it and get the identity, we can then, for example, calculate the biomass of copepods, krill, and other organisms we have in the nets. So that's a lot of the work that we are doing. And here I just quickly want to show you the results of this investigation, which actually shows that in the different regions we focused on, the Cape Verde Ocean Observatory, the Central OMZ, and the station at 5 North, we find different intensity of this diel vertical migration. And we only find strong diel vertical migration here at the Central OMZ. And we also see that we have a much higher zooplankton biomass, for example, at this location. And I think that this also is related to the fact that zooplankton in this region here, actually, as I said, that the oxygen tolerance threshold of zooplankton is lower than the environmental oxygen levels. So as I said, 2.3 kilopascal for the krill that we have investigated. The copepods even have lower threshold. But for example, the billfish that I talked about, they have a threshold of 7 kilopascal, which actually is above here. And as you've seen before, they are probably unable to migrate to below 300-meter depths in this regions anymore. And that actually might be one explanation why we find more zooplankton in this region and why we find more migrators in this region, because they might show higher survival rates due to lower predation pressure. Okay, so that was my talk. I would like to give you a quick summary. So a few main points that I wanted to make. Mainly, marine organisms have different oxygen tolerance thresholds, like we also. So some people can hike up on high mountains easily and others don't. And that's the same for different fish species and crustaceans. And so that is actually, in my view, very important to understand and to also further investigate, as the different thresholds actually also enable the organisms to act in different ways in these ecosystems. What we find, for example, is that off Peru, the krill conducts daily excursions into anoxia and uses the OMZ as a refuge. It's really a key strategy of these organisms, which enables them to build up extremely high biomass and for them to be really a keystone herbivore and a keystone species in this ecosystem. Tropical Atlantic zooplankton is less tolerant to low oxygen levels, something like 2.3 kilopascal oxygen partial pressure, but that at the moment is still sufficient for them to migrate down to the oxygen minimum zone and to use the oxygen minimum zone as a refuge in the tropical Atlantic as well. But if further the oxygen levels decline further, we might generate ecosystem-wide perturbation. So if these organisms might not be able to migrate further down, they are higher up in the water column and they might be vulnerable to other organisms preying on them, for example. And also this carbon export to depths, which I'm interested in, it might be impacted by lower oxygen levels at midwater depths. So this could declining oxygen levels could have significant consequences for food security and the carbon cycle. And so that's why we also try to do sustained observations in the tropical Atlantic. That's one goal of the MOPCA project as well. Yeah, with that, I would like to thank you for your attention. I would also like to thank a lot of colleagues who helped with this work, especially Lars Stemmann, with whom I work since 10 years now, deploying the underwater vision profiler, but also many other colleagues mentioned here who made all this work possible. Thanks for your attention.
Great. Thanks a lot, Rainer. I'm really impressed with this, and I'm wondering why we didn't have you in the biodiversity section. Or do you think of yourself as more thinking about the physical parameters and using the organisms just as sort of metrics of what's happening with the ocean physically?
Yeah, the main reason is that the techniques we use are not. So I see myself more as a plankton biogeochemist and biological oceanographer. So I'm more interested in the processes and the elemental fluxes, carbon flux, and these things. And the other reason is is this one that is, or rather this one, that is that we only that I'm taking images of the organisms in the ocean, and from an image you rarely can get to species level. Which is why I'm not working, why I think this tools are not perfect for biodiversity research. I mean, you can work on a broad level on biodiversity with also this data, and we to some extent also do that. And I think the future will be amazing. So what I'm thinking, what I want to do next also is to combine imaging and genetics to get to something that I would call imagenetics, where we would pick organisms from this scan and each organism and actually, for example, barcode it and get and thereby get the species ID, and then we can actually combine the benefits of the imaging approach, which, for example, gives us the area, it gives us potentially also the stage, or even maybe something like whether it's in a feeding mode or not, and we can combine that with the species information and can work on population dynamics and things like that. Yeah, so. Yeah, in short, I don't see myself as a biogeochemist.
Yeah, which is somehow really still in the domain of understanding biology from my perspective, but you know, that's what I do. So yeah. But I'm confused a little bit about, so maybe I missed this at the beginning. You said that the oxygen levels are declining, but why? Is that because of temperature? Mostly driven by temperature? You said that especially the top photic zone is low in oxygen, and is it also because of organisms dying and we don't have as much light, like photosynthetic organisms alive in the photic zone? Or
so the photic zone is normally the mixed layer, or where the water actually is turned over and is in contact with the atmosphere, and so the top layer of the ocean normally is oxygenated. So let me just go to yeah, this one, for example. No, I got out. So what you see here is the profile of Peru, and you can see that actually in the top 50 meters or so, you have oxygen, and that is due to the fact that the top layer actually is in contact with the atmosphere, and that the oxygen is replenished by the contact with the atmosphere, but also because, for example, phytoplankton produces oxygen. And then at depths, we have the oxygen minimum zones. And off Peru, it's very severe. There's really no oxygen. In other regions, it's less severe. And yeah, that we have this oxygen minimum zone here is due to the fact that the water here is very old. So that is transported from this water might actually, this water might even be 500 years old or something. So it went through all the oceans towards this location. And while the water is moving through the ocean, you also have the productivity raining down into this water parcel. And so while the water moves, the oxygen is continuously declining. And then in these regions, you also have high productivity. So there, this effect of oxygen removal at depths is even stronger. So that the mixing really can bring the oxygen down to depths, enough oxygen down. Yeah.
But I missed the part about why is it getting worse?
No, okay, sorry. I mean, warm water dissolves less oxygen.
Mm-hmm.
And so that is the main reason that actually it's just physical. The physical reason is like, let's say 50%, that if the global ocean is also warming, and therefore it's actually not so much oxygen is dissolving in the water that is formed actually. And the other reason is also that to some extent also warmer temperatures mean that the water is more stratified. So if the surface temperatures are higher, then the water itself is more stratified, which also means that there's less mixing in the water column. Yeah, so that's the two physical reasons, but there might also be biogeochemical reasons that actually we have changes in productivity, which then act on the consumption side. So much is on the supply side, but this consumption side is not so clear yet. Yeah.
Wow, cool. So I don't see any questions. Does anyone in the group have a question for, Nuria, please?
Yeah, hello. Very nice talk. Very nice. And also, could I say one thing, that you see yourself more in the biogeochemical part, but also I think that you have, your profile is, it's hybrid, that also you will fit very nicely in the biodiversity, that maybe, you don't identify the species, like in a very high resolution, or in a coarse way, but, or let's say that also we are a very, like a diverse group, and it's like that. The subjects that we are tackling, it's, they are general. That,
of course.
That you fit very nicely to everywhere.
Yeah, that's a fascinating thing about this data, that you can tease so much out of it. And that's also one of the main, my other focus is also to really generate this all in digital fashion, so that it's also useful for future generations, so to say. So, yeah.
Okay, and I have a couple of observations, and for example, here in this plot, that around 600 meters, we can see that there is an increase of oxygen, and is this due to the, I don't know, to the currents that they are coming from Antarctica?
Yeah, to some extent it's related to circulation, but it's also related to this interaction of what I've said, that you have on the one hand the supply of oxygen, but on the other hand also the consumption of oxygen due to material that rains down in the ocean. And to some extent the material is formed at the surface and it rains down in the ocean, and it's consumed while it rains down. And that means that you have also high consumption in these areas here at 100-meter depths, 200, 300 meter, and that the consumption of oxygen declines with depth. And then at some point, actually, we get to a state where actually the oxygen consumption is no longer so high that it completely outbalances the oxygen supply, which is coming with the ocean currents at this depth. And the other aspect that also explains this is that actually temperature actually declines with depth, and when temperature declines, then oxygen consumption is also getting lower. So that's also just a biogeochemical fact that actually at lower temperatures, biogeochemical reactions happen slower, and therefore respiration at depths in the deep sea is also less, because there's just the temperature effect. Yeah? So it's less material that is available at these depths for respiration, and respiration itself is slowed down due to temperature effects. Yeah. So that's the main explanations why the oxygen levels come up again, so to say. It's less that there's a lot of supply. It's more that the consumption side is slower.
And being of Peru, for example, how this is related, or when El Nino conditions applies, how the oxygen profile change from 1 year El Nino year and 1 year like normal conditions.
Yeah, El Nino is actually somehow it's counterintuitive. You would think that actually oxygen levels get even worse than during normal years, but they actually get better. It's not the oxygen that is a problem in this case. It's actually temperature. So what El Nino does is that warm water from the Central Pacific is actually transported to the Peru region, and that means that actually the thermocline is going down. So the area of cold water is actually suppressed. We have warm water on top of the cold water, and this warm water actually slows down the upwelling. And this means that less nutrients come to the surface, and that less productivity is happening in the area. And that means that actually there's less food. And so the dramatic consequences of an El Nino are due to the low food availability in the region, and not due to low oxygen. Actually, the oxygen levels are higher at this time in the top 100 meters than they are during normal conditions. But there's no food, and that's why there are seabirds dying, and low fish abundance, or low fisheries yield during El Nino conditions. Yeah.
Okay, thank you. And could I ask you, this is maybe like a kind of, I don't know, your vision? Like, for example, I am working mostly in coastal habitats. And do you think that coastal hypoxia is going to be, an important issue in the near future?
Yeah, I mean, that's already an important issue that is caused also by other impacts. A main impact of coastal hypoxia is also the supply of nutrients, for example, via rivers. And that also generates huge low oxygen regions and huge regions where actually fish abundance is reduced. For example, also in I mean, we have the Baltic Sea, where actually a lot of nutrients have been supplied in over between, let's say, 1970 to 1990 or so, and so the oxygen status of the Baltic Sea declined dramatically during that time, and then actually wastewater treatment plants were built to actually avoid the influx of nutrients from the rivers, and that actually had an impact. And the same is true for many river deltas, that they actually, you have a lot of that they are also regions of strong productivity, which then also due to the nutrient supply, which then also is converted to a coastal hypoxia. Yes. And this will also, I mean, the principal mechanism of, okay, less oxygen in warmer waters, that is a strong driver, and that is something that will go hand with global warming. So global warming is causing also ocean deoxygenation, and it will also cause further increase of coastal hypoxia, but nutrient input by human activities is another main reason for that.
Mm-hmm. Yeah. Okay, thank you, Rino. Yeah.
Fabulous. I still think, so you're doing your, it looks like from this slide that you're characterizing your biology, so to speak, by size, whatever's coming through the net at those different size levels. Is that how you characterize stuff? But then you also talked about different groups. So you were talking about, for example, the krill and the copepods there. So at some point you're actually going through those samples and putting them in bins of some kind.
Yeah, of course.
Yeah.
I mean, let me go back maybe, or maybe go to this one. Yeah, so this is the net catchers we get. So it's we use a 200 micrometer mesh size, and the opening of this net is 0.25 meters, square meters. So we get organisms in the size range 200 micrometer to something like 5 centimeter, maybe 10 centimeters. And what we then do with the whole sample that you can see here at the bottom, we put that on scanner, on a scanner, and then you can see here on the top right that you can actually distinguish different organisms. So this is a calanoid copepod. Here we have an appendicularian. This is a nuclei of a copepod. And I mean, we do image classification of all these organisms. So we actually, for each net, we take, so to say, we take digital images of a representative fraction of all the organisms. And then we get the size information and the identity, and we use that to, for example, calculate the biomass, but we can also calculate respiration rates then based on that, and many things.
So you're calculating biomass instead of actually weighing them, or you're doing both?
No, we don't weigh that. We calculate it from the images, and I think that's a better approach, because what was done before is that actually this sample, for example, would have been just taken, everything would have been crunched, and it would have been weighed, and then you get the weight of the sample, but you have no idea who was there.
Who is there?
Yeah, and how big they were. So, for example, it's also very crucial to know how big the organisms are in your sample, because, for example, if you compare elephants and mice, elephants have, for example, much slower turnover rates than mice have. And so the elephants and mice have very different roles in the ecosystem as well. So that's why, and this is also size dependent. So we really are also very much interested in the size, because size is a master trait in many relationships that we are interested in.
Yeah, cool. But when you report the results, you don't break it down by how many different types of organisms or species, or so that's what I'm getting at with the diversity thing. It seems like a really rich data set for, even if it's not exactly to the specific species, you have groups of species, and it seems like a hugely rich data source in terms of understanding how these profiles vary over the ocean and how they will vary in response to different temperatures and whatever's fluctuating from year to year in the climate. And I mean, it's just really awesome for understanding what's happening in the water column in terms of diversity.
Yeah, no, I mean, yes, we also discern it by different larger groups, or can also go to compare what krill does to what copepod does. And with the camera, for example, we also can identify especially fragile organisms which are destroyed in the net. So we do work on these questions as well. And for example, the nice thing about these net samples, or the digital work with the net samples, is that the samples are not destroyed. So as I said before, everything was put together. You had to get the weight, the biomass of zooplankton. You actually have to dry it. and that means you destroy your sample.
Right.
The digital technique is, or the scanning technique actually means the samples are intact afterwards and everything is still there. So we have these, all the samples that I've taken with this technology are still existing, and we will actually put them into a museum. And I hope that at some point someone will also go through the samples and actually go to species level with this approach. And yeah, I think in future we'll actually do image genetics to get really to all the things that are interesting. Yeah.
That's so cool. I had no idea you were doing all this cool stuff. Awesome.
Mm-hmm.
So I did have one other question about you had that story about watching the ship that was trying to find your probe or buoy. What did you call it? I can't remember, but your camera underwater. So all of your images are stuck inside that camera. They're not being uploaded somewhere in cyberspace. I guess that would be hard in the middle of the ocean out there.
Yes, the problem with that is, I mean, we get particle counts from this deployments. So this is called a BGC Argo float. You can see it here on the right, and you can see here the camera attached to it. And the principal data like temperature, salinity, oxygen, we measure chlorophyll A, we measure turbidity, and we've measured the particle abundance. So that's another thing. That's what the camera also can do. Like it can measure the particle abundance in different size classes. And this information we all get by satellite, but the individual image data is actually too much. And so we cannot transfer it by satellite, and that's why I'm really thrilled to get this float back, because now we can actually download it. What will be the future here is that we actually have the camera itself identify the images. So we'll do some deep learning embedded classification. So the camera will so to say do the job of identifying the organisms and particles and send us some data, some aggregated data. But of course, getting the images itself back right now is really very great thing. So I was very happy last Wednesday.
Cool. And you got it from all of them or just the one? Is this, you said there's 6 different ones here?
No,
it's just one camera.
This is one camera. We have actually 3 deployed. I hope to get one more back now in May, and I'm afraid we'll lose one because that one is going in the Southern Ocean, where rarely some research ships, or there are not many research ships there, and very much of ocean. So it's unlikely that a vessel will just pass by it. So it's very difficult to coordinate that. Research vessel per day costs 20,000 euro or something, maybe even more. So they will only do it, get it if it's on the track. So in this case, we were lucky that the vessel was just in place and wanted to go along that route anyhow, so they had to go this way, and so it was quite good.
Oh my goodness, talk about risks in research. Camille, did you have something to add?
Yeah, I was wondering, you mentioned that the warmer temperatures are already correlated with lower fish catch in some regions, but it's obviously very complex. It's not just a straight matter of the oxygen level, it's how that interacts with prey abundance and ability to forage for the prey at different depths. Are you comfortable in, how comfortable are you in being able to project losses of fish catch with warming sea surface temperature, given how complex you're in your presentation, you've shown how complex it is.
I mean, the example of El Nino is well known. So that's where I said, okay, warmer temperatures are related to less fish productivity, and that's a well-known example and problem. I mean, for Peru and Chile and this region, it's really a very strong ecosystem effects which are well researched. So in that case, we can definitely say that warming has such an effect, because all the physics and everything has been researched. How it works in other regions, I mean, that's why I'm working mostly now in the tropical Atlantic, is less well known. And of course, we also don't have, what the future will bring is to some extent unclear. We can model it, but we also have to observe it. And that's maybe also something. I mean, the changes in temperature that we expect are much faster than what we have seen in the past. So we actually also need to get faster with our observations. Yeah, and maybe as another example from Peru, there's just been a paper published this year where actually colleagues have looked into sediment records. So they have looked into you can take sediment cores from the seafloor and just have a look what you find at different depths and in the sediment, and these different depths then relate to different times in Earth's history. And so they looked into a time that was the last interglacial maximum, where also temperatures were very high in comparison to now, or similar to what we expect. And they found that they found no anchovy. So the main fish you find off Peru at this time, and so they found more gobies and other stuff, and so they also see in this paleo record that actually the fish community changed dramatically or was very different to nowadays in that area.
Yeah, it strikes me that it's enormously more complex than projecting changes in terrestrial or even freshwater species. Because, oops, sorry about that. Turn that off. Because in terrestrial systems, I mean, you do have an effect of CO2, which on top of sort of temperature, climate space.
Mm-hmm.
But in the marine systems, you've got not only the temperature, but the acidification, now the oxygen, and foraging, and I knew about the diel movements of species, but it somehow, when you were presenting it, it really struck me that they really are physically moving a lot between foraging areas, refugia, much more so than I think a lot of terrestrial species do. And so the whole story, I just, our lab does some terrestrial species modeling, and I'm pretty comfortable with just the climate space. It's not perfect. It's not everything. Yes, we know there's more complexity, but it's not bad. Mm. And I'm thinking in marine systems, it might, it just seems to me you've got a lot more complex a situation, even to get, an approximation of what's going to happen. Am I thinking about this right, or am I being overly pessimistic?
No, I also agree that there's a lot of work to be done. So if you consider the models, so the models for the IPCC report mostly contain the biogeochemical models that try to model the oceans, for example, oxygen distribution, but also the biomass distribution of organisms. They normally contain nutrients, one type of phytoplankton and one type of zooplankton and one type of detritus. And that's at the moment, or maybe 5 years ago, that was the level of complexity of these models. And of course, everyone in the community is working to get these better, which means we need to know better what the organisms are doing at different conditions, and how we have to actually model the different compartments. The zooplankton compartment, I mean, the zooplankton in these models is mostly a copepod, but we know that they are gelatinous organisms, and that we also have, for example, phytoplankton is not one type of phytoplankton. We have nitrogen fixers, we have small, we have large. So these models are good, but I'm sure that our picture of everything will change, and to add somehow to your confusion and to the complexity, I mean, we are also talking about water movement. So you might be able to observe a tree every year, whereas we have to the water at a location one day is different to the next day. So we also have this like movement of the water that makes things, and mixing of the entire ecosystem, so to say. I mean, the entire habitable space of zooplankton is constantly changing. So that's really making things very complex and very interesting. Yeah.
That's true. Scientifically interesting.
Yeah.
Wonderful. Well, that was a really fabulous seminar. We have one comment in the chat. In terrestrial systems, I guess key factors are well mixed. Oxygen, carbon dioxide, temperature, and much is in a 2D space, which is quite different in the sea.
Yeah, that's what I just said. That I mean, we have a 3D environment that also is constantly turning around. And for example, the diavertical migration, that's really huge and happening everywhere. It's actually the largest biomass migration on Earth. And if you consider a copepod or a krill of like, let's say 2 centimeters migrating every day 300 meters down, 400 meters, that's actually huge. And it's really something that also then is important for the biogeochemistry of the ocean. Yeah.
Oh, wow.
And if I may add, it's like that, no, Rainer, please correct me, that, okay, you have been mainly working or mainly, you know, mentioning plankton, no? So plankton in this case, not that it's a mobile component of the sea, but then there is all the fixed, let's say, component, let us say fixed, no? That are all the sessile organisms, no? Like corals, kelps, seagrasses, and so on. That it is, I agree with you, no, Camille, about this complexity and also this 3-dimensional, no? The depth gradient, that it's also very important.
Great. Well, thanks a lot, Rainer. That was pretty fascinating, and really is nice that it's crossing a lot of the different domains that we've been talking about in MOPCA. So thanks a lot.